We tend to think of our DNA as this fixed blueprint, right? An ancient instruction manual that's passed down, pretty much unchanged from one generation to the next. But what if that manual was constantly being rewritten? In this explainer, we're going to pull back the curtain on the surprising truth. Our genome is a living, breathing document. And the editors? Well, they're not who you'd expect.
For the longest time, this was basically the central rule of genetics. We imagined our genome as this sacred text, meticulously copied and handed down from parent to child, almost perfectly intact. It was the blueprint that made you, well, you. End of story.
But what if that whole story wasn't quite right? What if our genetic code was less like a stone tablet and more like, say, a Wikipedia page, with new sentences being added, cut, and pasted all the time?
Well, it turns out that's exactly what researchers have discovered. The old idea of a static genome is out. The new reality is a genome in a state of what's been called incessant flux. New genes are constantly being brought on board, creating this dynamic, ever-changing genetic landscape. So, if our DNA is being edited, who or what is holding the pen?
All right, let's meet the primary agents behind this genetic rewrite. We're going to look at a few key players, but the first one is a real renegade, and it's hiding inside our very own cells.
They're called transposable elements, or TEs for short, but honestly, they have a much cooler name, jumping genes. And that's literally what they are, segments of DNA that can up and move from one spot to another right inside our genetic code. And they are not some rare little anomaly.
And this is where the sheer scale of this just becomes kind of mind-boggling. Take a look at that chart. A staggering 44% of our genome is made up of just one type of jumping gene, with another 3% from a different kind. Just let that sink in for a second. Nearly half of what we call our human DNA is actually made of these restless, mobile pieces of code.
So how do they actually do this jumping? Well, they've got two main methods, and it works almost like a word processor for your DNA. On the left, you've got copy and paste. This is what retrotransposons do. They make an RNA copy of themselves. Think of it like hitting Control-C, which then gets turned back into DNA and pasted somewhere new. And this actually increases the number of copies. Then, on the right, you have the more direct cut and paste. The DNA element is literally snipped out and moved to a totally new location.
But hey, jumping genes aren't the only editors messing with our code. Bacteria can pass genes directly to other organisms, including us, in a process called horizontal gene transfer. And then, of course, you have viruses. They are absolute masters at hacking our cells. It's how they survive, by injecting their own genetic material right into ours.
And the impact of viruses on our DNA is, wow, it's staggering. A full 8% of the human genome is made of something called endogenous retroviruses. These are basically fossil viruses, the leftover remains of ancient infections that hit our ancestors' reproductive cells, their germlines, and just got stuck there. They became a permanent, heritable part of what makes us human.
So all this constant rewriting of our DNA has some pretty profound consequences, and it's really a double-edged sword, acting as both a destructive force and an incredibly creative one.
Let's start with the dark side, because there definitely is one. When one of these jumping genes lands smack dab in the middle of a critical gene, it's like throwing a wrench into the works. It can totally disrupt its function. This process, called insertional mutagenesis, is linked to some devastating diseases like hemophilia, Duchenne muscular dystrophy, and it can even create a predisposition to certain kinds of cancer.
But, and this is a huge but, here's the incredible twist. This seemingly chaotic and destructive process is also the engine of innovation. The very same mechanisms that can cause disease have also been responsible for some of the most profound creative leaps forward in the entire history of life.
So let's explore that creative side. We're going to look at a couple of incredible examples of how these genetic hacks actually built the complex biology we see all around us and inside us today.
This might be the greatest story of gene swapping ever told. Billions of years ago, a simple cell swallowed a bacterium, but instead of digesting it, the two of them formed a kind of partnership. A massive transfer of genes from that bacterium into the host cell's nucleus cemented the relationship, and that little bacterium eventually evolved into the mitochondria that are powering nearly every single cell in your body right this second. But what if I told you that one of the defining features of being a mammal, giving birth to live young, was actually made possible by an ancient viral infection? I know, it sounds like something straight out of science fiction, but the evolution of the placenta is maybe the most surprising innovation ever driven by these genome hackers.
Okay, so meet syncytium. This is a gene that is absolutely essential for forming the placenta. Its whole job is to fuse cells together, creating this crucial barrier between a mother and the fetus. And where did this incredible gene come from? It's a repurposed or domesticated gene from a retrovirus. It used to make the virus's outer shell.
And get this, it wasn't some one-off fluke. This timeline shows it happened at least twice in our history. About 40 million years ago, a viral infection gave our primate ancestors the gene for syncytium 2. Then 25 million years ago, a totally different virus gave our great ape ancestors syncytium 1. Our very evolution was hijacked by viruses for our own benefit.
So, if nature has been using these incredible tools for millions of years, well, the next logical step is for us to figure out how to use them too. And that's exactly what's starting to happen in the world of medicine.
So in gene therapy, the biggest challenge is, how do you get a healthy gene into a patient's cells? Viruses are natural experts at this, but they have risks. They can set off an immune response, or worse, insert the new gene in a dangerous spot. The cut-and-paste jumpers, the DNA transposons, they'd be a much safer delivery vehicle. The only problem? The ones in our own bodies have been dormant for millions of years.
So scientists did something amazing. They basically became molecular archaeologists. They found an ancient transposon in the genome of fish that had been inactive for ages. It was sleeping. By comparing a bunch of different fossilized versions of it, they were able to piece together and reconstruct the original, active version. They called it Sleeping Beauty, and it has become an unbelievably powerful tool. And this isn't some far-off fantasy. The Sleeping Beauty system is now being used in clinical trials to develop real treatments for a whole host of genetic diseases, from hemophilia to sickle cell anemia. We are literally harnessing an ancient genome hacker, resurrected from a fish, to fix our own DNA.
Which really brings us to this powerful quote that just perfectly wraps up everything we've been talking about. Our genome isn't this single, pure lineage. It's a beautiful, complex mosaic of our own ancestral DNA, all mixed in with genes from bacteria and ancient viruses that have been stitched together over millions and millions of years.
And this all leaves us with a really profound final thought. If our DNA really is a mosaic, part ancient human, part bacteria, part virus, what does that change about how we define ourselves? It suggests that we aren't these isolated individuals, but living, breathing collections of life's entire history, far more connected to the great web of life than we ever imagined.